right? And this is all about diffusion and osmosis. And at this point, you've got a pretty good sense. What is diffusion? If you at your elevator talk, somebody is like, oh, interesting. What is diffusion? Could you tell me just in a nutshell?
So moving from high concentration to low concentration.
Right, so there's some like usually molecule or ion or something. It's moving from high to low. What other things could you tell me about it?
Hydrogen ions are breaking off.
No. Hydrogen ions are not necessarily involved in this at all.
Facilitated or unfacilitated?
Right, so even before that, what's a more important defining quality of diffusion?
High solute is normally spreading to a lower solute.
But it's passive. You don't have to add extra energy to it. And the movement is always from high to low concentration. Osmosis, is just a special version of diffusion we talked about last time, but instead of looking at some solute moving, we're actually thinking about the solute being constrained by some semi-permeable membrane and the water moving instead. It's worth talking about some terminology that we use a lot about osmosis. Let me just do that briefly here. Let's start with a blood cell. What is the osmolarity of just all of the dissolved stuff inside a blood cell?
0.3.
Exactly, 0.3. You know that this is going to be at 0.3 osmolar, because that's basically almost everything in the body is at 0.3 osmolar. So let's think about putting this blood cell in different kinds of solutions. I'm going to have 3 cases. I'm going to have one where I'm putting it into distilled water, one where I'm going to be putting it into like 0.3 osmolar, and one I'm going to put it into something that's 0.6 osmolar. So these are different solutions of whatever. This is, well, this is just water, so it's not a dag nabbit. There's my water. This has something dissolved in it, who knows what. It could be salt or sugar or whatever. If it was salt, what is the molarity of the salt that would give me 0.3 osmolar?
Would it be 0.15?
Exactly.
Hmm.
Right? If I had 0.15 molar NaCl, because each Na and Cl becomes independent, we just double that. So that becomes 0.3 osmolar. Right? So 0.15 molar NaCl is the same as 0.3 osmolar. If it was glucose, just sugar water, what would be the molarity of the glucose to give me that osmolarity?
0.3.
Just be 0.3. They're the same. Exactly. What would happen if I had 0.3 molar NaCl? What would it become in terms of osmolarity?
0.6.
Yeah, exactly. 0.6. This could very well just be 0.3 molar NaCl, right? 0.3 molar NaCl dissociates into 0.6 osmolar because every NaCl becomes two independent particles. So that all makes sense? All right, so now let's take our blood cell and put it in these different things. So in this first case, again, we always start with like what is in the cell itself, and we know that within the cell is 0.3 osmolar. What is the osmolarity of distilled water? How many particles per liter do we have in total in pure distilled water?
0.
Zero. Right? There's nothing. There's no distilled water means there's nothing dissolved in there. So I have 0 outside and I have 0.3 inside. So whenever you have a solution that has a lower osmolarity than a cell, you say it's a hypoosmotic solution. So hypo meaning below, right? Hypodermic needle goes below your skin. Hypoosmotic. If I have, again, 0.3 inside, 0.3 outside, if it's exactly the same inside and outside, I call this an isoosmotic. Iso means the same. Right? Isometric exercises where you don't change the length of your muscles. Iso just means same. In this case, where it's more than inside the cell, it's a hyperosmotic solution. Hyper, right? Whoa, more. So hypoosmotic, isoosmotic, and hyperosmotic. This just has to do with comparing the osmolarity inside to outside. And the inside is always 0.3. So basically, if the solution is less than 0.3, we call it hypoosmotic. If it's exactly 0.3, it's isoosmotic. If it's greater than 0.3, it's hyperosmotic. And I should say, just for completeness, this 0.3 is somewhere between like 0.28 to 0.3. But let's just say 0.3 because it's easy. So that's one thing we care about. The other thing we care about is what happens to the cell. Is the cell, what's going to happen with osmosis? And so there's this other term, tonicity, that you need to know. Tonicity is kind of like the pressure. Which way does the water go? And in this case,
11 a.m.
In this case, you have to kind of remember tonicity is water moving rather, and it's going to be related to osmolarity, but it also is related to some other things as well that we're going to see in a few moments. So back to this picture, we are going to assume, let's assume non-penetrating solutes. So assume whatever is making the osmolarity inside and the osmolarity outside cannot cross the membrane, and they're locked on either inside or outside of the cell. This is not always a good assumption. We're going to, in fact, our lab next week, we're going to be working with some penetrating solutes like glycerol and urea. But for right now, let's assume, what's an example of a non-penetrating solute, something that can't cross the membrane without help?
Ions?
Say it again.
Ions?
Exactly, ions. Sodium chloride is a non-penetrating solute. What's another non-penetrating solute?
Glucose.
Glucose, a big polar molecule. So if we're talking about those kind of things that can't cross the membrane, then that's the situation we're in. So now let's think about tonicity, which way the water is going to move. If I'm in a hypoosmotic solution, less osmolarity outside compared to inside, which way does the water move?
Into the cell.
Yeah, the
Out of
water's
the
going to
cell?
move in. The water, remember I talked about kind of the salt draws the water in. So this is going to be the water moves in. This is called hypotonic. Water moves into cell. So depending on how strongly hypotonic solution it is, it might just kind of swell. If it's strongly hypotonic, it might be enough to pop. If the cell pops, we call that hemolysis. Lyse means to cut open. So hemolysis, cells break up. If it's really strong, that happens if you put your blood cells in distilled water, they all explode. They just spill their hemoglobin into the solution, and it just becomes this big red solution of free hemoglobin. There's no more cells in there anymore. All the cells have popped. What about if I have an isoosmotic solution where it's balanced inside and outside? What's the net movement of water?
0.
0.
Isn't it gonna go, oh, I thought it would go both ways.
Right, but there's no net movement of water. So water can cross, so water can move out and it can move in, but there's just as much of a tendency for water to drift out as drift in, because there's no difference in the osmolarity. So this is what we're going to call isotonic. Now I'll say no net movement of water. Because no, you're right, the water can come in and out, but ultimately you're not going to have more coming in versus coming out. So it's not going to inflate or deflate ultimately.
I have a quick question.
Uh-huh.
So when you're using terms like hypotonic and isotonic, is it only relating directly to water, or is it just any type of movement of fluids through the membrane?
No, it's the movement of water in or out of the cell.
I have a question.
Uh-huh.
So this would not be oscillating back and forth, right?
I mean, when I think of oscillating, I think of a more regular kind of bouncing. This is more just, random. It's like there's some party going on and sometimes people are coming in, sometimes people are leaving, and on average, you have the same number of people leaving per hour as are arriving at the party per hour. So the party isn't actually getting bigger or getting smaller. The people who are actually at the party are kind of changing, but it's not like some thing where someone's going in and out and in and out and in and out and in. That's when I think of oscillating, when there's some actual kind of bouncing going on or something. So it's more like or I don't know, a party, museum, the museum. Across the course of the day, people are entering the museum, people are leaving the museum. On average, there's always about the same number of people in the museum, but there's no net movement in or out. But that doesn't mean there aren't people entering and leaving. But overall, the balance is the number of people in the museum isn't changing. Same thing, the water, some water moves in, some water moves out. But as you just look at it over time, it's not inflating or deflating. So I put a happy face here. This is why we came up with our IV flush syringe to be 0.3 osmolar. It's isoosmotic and isotonic. And that means that when you shoot it up into somebody's vein, that the blood cells are going to have a little smile on their face because it's like, this is just what we like. So what about if you put this poor blood cell into these 0.6 osmolar?
It's gonna shrivel up.
Yeah, the water's going to leave. The water goes to where it's more salty. We're going to get what we call crenation, which it gets all kind of shriveled up. And we call this a hypertonic solution. So H2O leaves the cell. So this is like over the weekend, spend some time with these terms: isotonic, hypertonic, hypotonic. Again, that has to do with the movement of water. It's related to these hyperosmotic, isoosmotic, hypoosmotic. But as we're going to see, there can be things that make the relationship more complicated than what you see here. If it's a non-penetrating solute, then the relationship's super easy. It's hypoosmotic is hypertonic. I mean, hypoosmotic is hypotonic. Isoosmotic is isotonic. Hyperosmotic is hypertonic, assuming that the solute does not have the ability to cross the membrane. I'll show you the reason why it's not always true though. Like assume a penetrating solute. Like for example, urea. Urea is small enough that it can cross the cell membrane without any help. So I'm going to take 0.3 osmolar urea. So this is isoosmotic urea solution. You know, I'll make the ureas little yellow things. Maybe I'll make them. Right, urea is a little nitrogenous thing. That's from the breakdown of amino acids and stuff. So we have, 0.3 moles of urea per liter. It's just a concentration of urea molecules floating around in there. I'm going to put my blood cell in there, which is also 0.3 osmolar. So here I have an isoosmotic solution, but it's a penetrating solute. So the moment this hits the solution, maybe for a fraction of an instant, maybe things are okay, but after the first instant, things are not going to be okay anymore. Because there is not urea inside a cell normally. Normally, urea is not inside a cell. It's a waste product that you get rid of. So how much urea is in the cell to start with?
None.
None. There's no urea. But there's a whole bunch of urea outside. So I have a whole bunch of urea outside, no urea inside. Which direction does the urea want to go if we just look at the diffusion gradient of urea?
Into the cell.
Yeah.
It wants to go into the cell, high to low. It's 0.3 out here, 0 in the cell. So urea wants to come in, as did the glucose or the sodium chloride, but they couldn't because they couldn't cross the membrane. What happens though with urea, because it's penetrating, it wants to come in, and by Jove, it can. So the urea crosses the membrane and the urea starts coming into the cell. What's going to happen to the osmolarity inside that cell as I start adding more solute in addition to what's already in there?
Gonna go up.
Gonna go up. Starts going up. And so now all of a sudden I have a higher osmolarity in here. So maybe this is going up to 4 or 5, whatever. It's still 0.3 out here. What's water going to do now as the osmolarity in the cell starts going up?
It'll want to go in, right? Or
It's
out,
going
in.
to want to go in, right? It goes to where it's more salty. So urea enters, the water follows. Because as the osmolarity is increasing in the cell, the water is going to come in by osmosis and kind of dilute it. What's going to happen to the cell now?
Hemolysis.
It's going to start blowing, it's going to start inflating and probably hemolyze. So so this is an isoosmotic solution. What is it in terms of tonicity? If water enters the cell, what do we call that?
Hypotonic.
Hypotonic.
Yeah, exactly. So remember, hypotonic if water enters the cell. So here we get this kind of fascinating thing. This is hypotonic. I should do a larger. It's hypotonic even though it's isoosmotic. Right, so that's where you have to be careful. In our lab next week, this becomes important because we have a couple of penetrating solutes we're going to be working with. So this idea of a solution, and again, obviously it makes sense to understand the details of what can you put in somebody's body and not explode their cells, right? This is all about what kind of a solution or how a particular solution is going to affect your body cells. You have to think about two things. If I have a red blood cell and I have it in some solution, and I want to know what's going to happen to this puppy, what are the two things I have to think about?
Osmolarity.
I have to think about osmolarity. Is it greater than 0.3? Is it equal to 0.3? Is it less than 0.3? That's going to make a big difference. And what's the other piece of information I need to make a definitive prediction?
The tonicity.
How do we figure that out?
If it's penetrating or not.
Yeah, is it penetrating or not? So these are two things that you are going to need to pay attention to when you are trying to predict how a cell is going to react in some solution. And then you have the information you need, the stuff we've just been talking about. If it's non-penetrating, then it's easy. If it's less there, then if it's less, then it's going to be hypotonic. Water is going to come in. If it's equal, it's isotonic. If it's greater than 0.3, it's going to be hypertonic. Water is going to leave the cell, it'll shrivel. If it's penetrating, then you got to be careful because it can even be isoosmotic, but still be hypotonic because the penetrating solute enters the cell and then the water follows, which ultimately ends up inflating and bursting the cell. So does that make sense? So in fact, I can just, I'm going to share. modules. So this is something you just have to do before next Tuesday. You got all weekend and the beginning of next week to do. This fluid balance and RBCs lab. First there's the Prelab. There's a little thing that I made that basically talks about what I just talked about, to review it again. I gave you a little PDF thing, which it'll be, don't actually print this out and type it in. You can make this chart yourself in your lab book, but basically we're going to have different kinds of solutions, and you have to predict what's going to happen to the blood cells when they go into these different solutions. So this PDF here is just to try to hold your hand as you start thinking about this. Does it dissociate? If it does, into how many particles? Therefore, what is the osmolarity of the solution? Like, you've got the molarity, but you want to know what is the osmolarity. It's going to relate to the dissociation. And then, compare. Is it hypoosmotic, isoosmotic, hyperosmotic? It's just going to be comparing it to 0.3, right? So this is just what I've been talking about. Then you got to think, is it penetrating or non-penetrating? Just as a spoiler alert, salt and sugar do not penetrate. You know that already. Urea, I just kind of told you it does penetrate. Glycerol. Glycerol is like an alcohol, which does have it has hydrophobic and hydrophilic properties. So glycerol typically does have an ability to get into hydrophobic domains. I'm going to leave the ammonium chloride as let you figure that out. That one is kind of, it's an interesting thing. I'll talk more about it after we do things. But your prediction, what do you think is going to happen? Is the cell going to swell or hemolyze, no change, or is it going to shrivel and crenate? So this, as you come up with your hypotheses, it's going to just be what I've talked about. You're going to have to think about the osmolarity of the solution and how it compares to 0.3. Also worry about if it's penetrating or non-penetrating, and make a prediction. It shouldn't be that tricky once the concepts are clear. But this is going to kind of force you to really think about the concepts to get them clear. Why, yes, Ms. Woods.
But it doesn't make a difference if something is regularly occurring in a cell versus not. Like with urea, we looked at it. Urea, you said, isn't naturally occurring in a cell because it's a waste product, and therefore it's going to rush in. But isn't there an equilibrium if there's already some in the cell?
So what I would say is that 0.3 is resulting from lots of different things. So any particular individual thing that you're thinking about is going to be much less than 0.3, because it's just one little piece that contributes to that 0.3 total moles of total particles. Okay. So yeah, any individual thing you think of is going to be way less.
Thank you.
So I just talked a whole
I
bunch.
have a question real fast. How do you determine if something is penetrating or not penetrating?
So you tell me, what were the qualities of things that could penetrate a cell membrane without help?
It's non-polar, it doesn't need help.
So non-polar.
Small.
And if it's polar, then it's super small, right? So again, that's what I was just talking about. Urea is polar, but it's dinky. And it does have an ability to kind of jam through the membrane sometimes. Glycerol, I said, is an alcohol.
Where that document was that you just showed us?
What's that?
Where is that? I
can't find where that document you just showed us was, that chart.
Let me make sure that it's shared properly. So if you go to lab, fluid balance and RBC pre-lab, and it should be visible. And then intro to fluid balance and RBC lab and pre-lab guidance. So that should be there. And if you click on that, it should have the stuff that I just showed you.
I see it,
And
thank you.
then later today, I will activate the submit PDF of fluid balance and RBC pre-lab, which is just going to be your objective and hypothesis. Right, in your objective for this lab, what are, just from what I've been saying, given that we are blah, 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 blah. Hold on, where's my, oh, I didn't. Now I want to go back to this table. You can see what kind of things are we changing the independent variable in the different solutions.
Similarity.
One is we're changing concentration, exactly. We have different concentrations of our solutes. And what's the other thing that is changing?
Penetrating and non-penetrating.
Yeah, just kind of the types of solutes themselves. So we kind of have two independent variables. One is different kinds of solutes, and then among those solutes, different concentrations. Right, and then our dependent variable is ultimately going to be what?
The solution.
No, the solution is,
Osmolarity.
no, that's going to be part of our calculations to figure out, but we're trying to,
The type of the red blood cell.
what's that?
What happens to the red blood cell?
What happens?
Hmm.
A dependent variable is going to be like the fate of the red blood cell. Does it explode? Is it happy? Does it shrivel? Right? So that's our dependent variable. That's the thing that we are going to make predictions about in the hypothesis and then actually check for when we do the actual experiment. So did that make sense? Yeah, so I think what we're gonna, for today's lab, I'm gonna go back. Let's go back again. No, let's go to modules. Modules. Let's just do this way. So Osmometer Lab. If you go into this doing the Osmometer Lab, there's just like a little video that shows me setting up the osmometer, and then just a 3-minute thing where I just talk about the actual like 20% versus 40% in their race. You know, this is something I don't have to like sit, babysit you while you watch. We can just think what we'll do. I'm going to just kind of spend the next few minutes kind of showing you this and then just kind of let you guys head into it. These two videos just show the setup of the osmometer and what we're doing, so you have a really good sense of what actually happened. And then here's this data. When you look at the data, oh, I gotta share, hold on. When you're looking at the data, make sure that you realize that there are tabs on the bottom, like here for osmometer. This is the raw data, the different groups what they measured in terms of the height, the kind of the absolute height after 10 minutes, 20 minutes, 30 minutes, 40 minutes. This was kind of the average height for across the different groups. Average rate is just how much did it move in that last 10 minutes divided by 10 minutes. So you get millimeters per minute. This is the 20% group, the 40% group. If you move over to height graph, this is just, I've pre-made a little graph showing you the two groups. The rate graph shows the rates for the two groups. So you don't have to make the graphs from scratch. You have the graphs actually here as a height graph and a rate graph that you can use. So that's the first part here. So all you're going to do for the Osmometer Lab stuff is look at these things. It's going to be like 11 minutes worth of videos to see what you're doing. And then you've just got the data, and then you have to kind of decide what does it all mean and write up your discussion on it. The other part, this doing the diffusion demos, we are not going to do it as much. So I'll tell you what you need to do. It's going to be a little different than what it says in the lab book. All right, so this is doing the diffusion demos. For the Brownian motion part, where it talks about the Karman dye and all that, we're not doing any of that. Instead, you're going to look at this little video of Brownian motion in pollen grains. So you're going to describe what you see in this little video and what is happening. This I will show you just briefly. If you take pollen grains and just put them on water and look at them under the microscope, what do you think you see? I mean, I did this when I was like a kid, I remember, because it was just cool. Has anybody ever done this? Nobody? All right, so what you're seeing here is just flow of the water, but what let's get to where, here's pollen grains. Can you see, what do you see?
They're moving.
They're wiggling and jiggling. It's under a very high magnification. But I'll tell you, pollen grains don't move on their own. So what do you think is happening? Why are they jiggling like that? Anybody have any sense?
Because the water molecules are constantly breaking and reforming.
So not even breaking and reforming. The water molecules are just constantly zipping all over the place. We've got all these little H2Os, which I'm kind of drawing as dots, going bam, 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 bam. There's just water molecules. So this is a way, you don't actually see this motion of everything that is responsible for diffusion, because we can't see actual molecules under a microscope, they're too small. But by watching the little pollen grains jiggle, imagine you have somebody standing in the middle of the room and everybody is shooting at them with tiny little BBs, right? Right, so they're sitting there and you just see them, you don't actually see the BBs, but you just see them getting hit one direction, another, forward, back. It's like, something's going on. You're not quite sure what's going on, but so this is the same thing. The little water molecules are just whacking these little pollen grains everywhere. And this is, kind of indirect evidence of this motion, kind of submicroscopic molecular motion that is going to be responsible for diffusion and osmosis. So all I want you to do is look at this video and really write down that you understand why this is happening. This was actually first figured out and described by none other than like Albert Einstein. He was the one who figured out that people knew that the pollen grains were dancing, but he was the one that figured out that this is evidence that actually of this kind of molecular nature or atomic nature of matter. And that water is actually tons of tiny little things zipping around and bashing into things. The other part here, part 2, this is, I talked about last time how molecular weight can affect diffusion rate, that if things weigh more, they have lower velocities for the same amount of energy, so they tend to diffuse slower. So this is like a little 5-minute thing where Tina will kind of guide you through the little experiment as you take, basically we're gonna, if I can show you really briefly. You know, it's basically taking these dyes made out of this purple dye that weighs like half as much as this blue dye that we're also going to be looking at. We got this blue dye, methylene blue, which weighs twice as much in terms of molecular weight compared to potassium permanganate, which weighs half as much. And you get to see how fast is it diffusing, you know, so she's measuring it at, you know, different times along. So watch this, it's like 5 minutes. When you're done, it says, note, the recorded data for this part is contained as a tab in the spreadsheet available in the previous osmometer module. I should have just put it here as a separate thing, but I didn't. So if you go back to that spreadsheet, that Osmosis Lab spreadsheet, and you look, it's like, oh my God, there's the agar diffusion tab. You know, this is just showing, here's the average distance that the lighter dye, the dye that weighs half as much, went twice as much in the same amount of time as the heavier looking one, or the heavier one. So again, you just have to kind of show how this supports this idea of molecular weight. So are there any questions about that?
Yeah, sorry, I'm just a little confused. So this is for today's lab. I guess I didn't realize what I was looking at was just the transport across the membranes, but this is like connected to the next part in the lab manual. I don't know if anyone else was.
Right, so if you, here, I'll show you. Let me go. If we go into our home here, for today, the official thing was osmometer and diffusion demos. Those were the 2 things that we were going to do in lab today.
Okay, I missed that part. I just saw the first part. Okay.
So what I would like you to do, again, I'm gonna just, I think it's, there's you can just look at it and do that. This doesn't need a lot of hand holding. So I'll just let you guys do the osmometer and diffusion thing. You know, sometime before Tuesday, you should come up with your hypotheses for this other one. And then there is no lab officially on that Thursday. That Thursday won't have, we'll talk about stuff, but we're not going to do anything. The one other thing I'll mention before we just leave for today. So osmosis and diffusion, they are critically important in everything that we're doing. But it's also really important to understand the limitations as well. So I like to use the example of, you know, the little old lady who's wearing way too much perfume. Like as you get older, all your senses get dulled and you think you're putting on just the right amount of perfume, but everybody else thinks you're putting on way too much perfume. And so this little old lady who's got tons of perfume walks in the door of the room and everybody else in the room is like, whoa, she's wearing a lot of perfume. So obviously the perfume molecules are making it up your nose. Like, how did those perfume molecules make it up your nose? How did they get from
Diffusion.
her? What's that?
Carriers.
Diffusion.
So you think diffusion, but it isn't. When you look at diffusion formally, diffusion only goes fast over a very short distance. Like the thing with the diffusion in that thing that Tina will show you, where you have the dye diffusing, like the dye went like a few millimeters in an hour, barely moved. And actually over the course of days, it would still be less than like a decimeter or something. Like when like if a cell was like 10 centimeters in diameter, if I had an oxygen here, it would take months before it would actually diffuse all the way to the center of this thing. So diffusion is great over a fraction of a millimeter. But once you get beyond a fraction of a millimeter, diffusion does not move things very far, very fast at all. So how is it actually that we get the oxygen to all our cells, given that, you know, here's the oxygen out here and all our cells are in here. How do we get the oxygen to them all?
Transport proteins?
Or even more, think about, so it's bound onto hemoglobin, which is going through your bloodstream, right? You bring the oxygen down into your lungs, then put it into your cardiovascular system, and you have literally 100,000 kilometers of blood vessels just streaming everywhere, carrying the oxygen through this river. And once you're in the cell, once the oxygen is near a capillary, you can then diffusion can do its job, move out of the capillary to the cell, but that's just a fraction of a millimeter. So this idea of convection, kind of the flow, that is a really important way things move as well. That's how your blood works. Blood flow, you pick up the oxygen, but then you flow in these fluid flows, convection flows, to bring it close to the cells, so then you can have diffusion, bringing it the last little way to the actual cell itself. When the little old lady walks into the room, there is all sorts of currents in the air that you don't normally see because air is invisible. But if you've ever, like, you take a drag off a cigarette and blow it into the room, it makes all these beautiful swirls and everything, right? It's because of all these currents and breezes in the room. That's what's carrying the little perfume molecules to your nose. If you took sugar and dumped it in your cup of coffee, let me do that. If you put sugar in your coffee and just put it in the bottom of the cup, you could wait a week and the top of the coffee would never be sweet, because diffusion doesn't move that fast. The very bottom would start getting sweet, but a few centimeters away, there'd be no sugar at all. So how is it that we get the sugar throughout the whole cup? Yeah, you stir, convection, you get the currents going and move it all around. So I do want you to kind of think about the kind of limitations as well of diffusion. Diffusion is super important. We'll see it all the time, but it will only move things across a very short distance in the period of time that we care about. So, you know, convection, kind of flow, is going to be also super important to move things around from one place to another. You know, particularly that example of the bloodstream, or even getting the oxygen into your lungs, this bulk flow of air, pressure differentials, and you get just a whole bunch of air moving in as a bulk flow, right? So bulk flow and fluid flow convection is another way things move. That's going to be super important as we go through the semester. Blah, 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 blah. All right, any questions here?
I know you said that we should just draw our own chart, but is it okay if we scale it down and do print it out just for like neatness purposes?
No,
for the
I'd rather you, I like the idea of you drawing it yourself, because that really makes you think about what is each thing and what am I writing. So yeah, I like people to write it in. 'Cause
But you would like us to print out the other like class data like we did before and put that in that we're analyzing?
Yeah, absolutely.
Do you want two different objectives, two different hypotheses for both part 1 and part 2 of the Osmometer Lab?
No, the diffusion demos are just demos. They don't have any kind of objective or hypothesis. You just have to describe Brownian motion and then describe the behavior of the heavier versus a lighter molecule when you look at diffusion. You know, and then interpret why that makes sense. Yeah, so there's no pre-lab or hypothesis or objective for those.
Okay.
Blah, blah. Where do we find that table? Which table? Nicole?
The one we can't print, right? You want us to create it?
Oh yeah, so that is, I was showing you all, that is in if you are in your modules, and you go down to the module for, you know, lab 4, fluid balance and RBCs, pre-lab, and go to this intro to fluid balance and RBC lab and pre-lab guidance, and click on that page. It's right here at the bottom. So I have a little thing where I'm yapping basically what I just yapped at you. And then there's a PDF of that very chart. Any other comments or questions there?
Yeah, sorry, back to the demonstrations. So as far as what we're writing up, if we just pretty much like on this paper, there are like questions. So as long as we kind of describe what we're seeing and respond to what the questions are on the paper, that kind of covers it.
Yeah, in fact, here, let me take a look at, let me double check here. So diffusion PDF, let me double check. Yeah, so for Brownian motion, you're not going to do any of this wet mount. Instead, you are just going to describe the video of the pollen grains. For diffusion, you are going to answer the questions here. And then these questions are worth answering. You should answer these because these are kind of making sure you understand like the main points that we're trying to illustrate by doing these demonstrations. So, you know, and especially after everything we've been talking about, this should be piece of cake. What's the difference between primary and active, primary, secondary active transport? You got it. What's facilitated diffusion? You got it. And if you don't, then this is a good time to go back and get it. So yeah, I would say part 3, you should be able to answer these questions and you just, you know, just write them down. So makes you feel confident, you know it, and makes me feel confident, you know it. All right.